Hi, everyone. Good noon. Um, Welcome to our DISL online seminar series, the third one. Today, our speaker is Dr. Bloodgood. She's a new research veterinarian. Did I spell that correctly?
Yes.
Yeah, research veterinarian working in Dr. Carmichael's lab, and she just joined us in July this year. So Dr. Bloodgood got her Ph.D. And veterinary medicine degree in University of Georgia. Her research focuses on the health of wildlife and using an interdisciplinary approach, incorporating social sciences and natural sciences ideas. So specifically, she studies the blood biochemistry, not geochemistry, the blood biochemistry in wildlife, also the metagenomics, and also studies the effect of the diet on green turtle health, as well as the influence of human activities on them. So she has wide collaborations with universities, statewide agencies, CDCs, etc. And so without further ado, welcome Dr. Bloodgood, and the screen is yours.
Okay, great. Hi, everyone, and thank you so much for having me speak with you today. I'm really excited about having started this new position as the research veterinarian with DISL in Dr. Carmichael's lab. And as a new employee, I really just wanted to introduce myself, as well as my previous research and some of the research that I've started and hoping to work on here at DISL. And I really wish that I could do this in person and meet everybody in person, but hopefully we'll get to do that sometime soon. So I'm originally from Georgia, and I completed both my BS and MS degrees at Clemson University. After that time, I did make my way back to Athens, Georgia, and worked as a research technician at the University of Georgia there. I then moved to North Carolina, where I worked at NC State University, as well as at the Museum of Natural Sciences, and eventually made my way back to Athens a third time, where I completed my dual PhD and DVM degrees on green sea turtles, which is what Sean was mentioning. And then just this past July, I started my position here at DISL. So now that I've shared a little bit about where I'm from, I really wanted to highlight some of my research experiences and talk with you about what motivates me as a researcher. So one of the reasons I'm really excited about my position at DISL is that it really lets me integrate all my areas of research interest and expertise. So I have experiences as someone trained in both wildlife biology and veterinary medicine. I also have experiences working in the human dimensions of wildlife health. And as I'm sure a lot of you are aware of, wildlife health investigation is an inherently interdisciplinary collaborative process. And that's something that I was specifically trained in through my PhD program, which I'll talk with you about a little bit later. And I want to emphasize the collaborative nature of a lot of the projects that I've worked on. And I definitely welcome any ideas for collaboration with you guys. So as I mentioned, I completed both my BS and MS degrees at Clemson University, and I had opportunities while I was there to participate in international research in both India and South Africa, including a summer field season at Bontebok National Park during my master's, which I will talk a little bit about shortly. But first, I want to share my master's thesis research, which focused on bald eagle egg lay dates in relation to climate change. This was a collaborative effort by Clemson University and Michigan State University through the Sea Eagle Network. And my advisor, Dr. Bill Bowerman, had a nearly 20-year data set of bald eagle hatchling data. So they did aerial surveys in Michigan and Minnesota, and they would identify eagle nests through the air and then employ a ground team to the nest tree. And someone like in this picture here would climb the tree, collect the eaglets and bring them down to the ground for morphometric analyses and other data collection. So my project was using the 8th primary feather length and sex data to determine the hatch date of the bald eagles. And then I compared hatch date over time in relation to climate change. So here you can see on the top chart, we have Julian date of hatch, which is basically just the day of year that the egg would have hatched. And on the x-axis, we have the year. And the top graph is showing that over this 20-year time period, bald eagles were laying their eggs about a third of a day earlier per year. The bottom chart is the same setup, but now with the average February temperature on the x-axis. And you can see that with every degree Fahrenheit increase in temperature, bald eagles were laying their eggs about half of a day earlier per year. And the most important takeaway from this data is that if species don't adapt to climate change the same way, there could be a synchrony between hatch date and food availability, which would then lead to decreased survivorship and fitness of the young. As I mentioned earlier, I also had the opportunity to lead some of my own field research in South Africa during my master's. And for this project, I photo documented family groups of Cape Mountain zebra and the prevalence of equine sarcoids. So sarcoids are a skin tumor that's associated with a virus and can actually be transmitted through fly vectors. And you can see an example of one of the tumors on the lip of this animal here. And because zebra can be uniquely identified by their stripes, much like how dolphins can be uniquely identified by their dorsal fin, I was able to photo document family groups and track the prevalence of equine sarcoids in their groups to direct management decisions on translocation and culling. These early research experiences really convinced me that I definitely wanted to work in wildlife health, and so I spent the next several years gaining some broad experience. As I mentioned, moved back to Georgia, where I worked at UGA in Susan Wild's lab on an avian vacuolar myelinopathy project. And AVM is caused by a cyanotoxin produced by a cyanobacteria that grows on aquatic plants like hydrilla, which you can see in the top left image here. And then coots and other aquatic birds will eat the hydrilla and thus be exposed to the cyanotoxin. And they develop a neurologic disease, and they'll start swimming in circles and things like that, which makes them very easy prey, as you might imagine, for predatory birds like bald eagles. The cyanotoxin then bioaccumulates up the food chain, and bald eagles very quickly will develop a neurologic disease and die from their exposure. So I worked on the project in its very early phases of toxin extraction and identification, as well as some experimental infection trials in chickens to prove that something on the hydrilla was causing the disease. I then, as I mentioned, moved to North Carolina, where I worked at NC State University. And for this position, I had great opportunities. I got to snorkel throughout the state, collecting endangered freshwater mussel species like the Tar River spiny mussel shown in the top image. And what we were doing was basically developing laboratory methods to be able to produce mussels to help stock endangered species. So the bottom picture is a picture of the Glaucidia, or the larval stage of the mussel, and we were basically finding which fish host species they prefer to attach to and propagating them in the lab. So that was a very exciting time. And then the middle picture here is me working at the Museum of Natural Sciences. And this is actually one of my favorite parts of my background, so I want to take just a second to talk about it. The museum is really where I learned that I have a passion for teaching and helping connect people to wildlife research and conservation. And it was also the beginning of my veterinary experience. So I worked on exhibit in the window on animal health, where I actually wore a microphone and interacted with the public to talk about the natural history of the animal that we were working on, and we would do everything from physical exams to surgeries inside the window. And I would also explain the veterinary side of any procedures that we were doing. You can see in the picture here that the public was right outside and they were able to interact with us and ask questions. I also had the opportunity to mentor multiple interns and students, and this type of work is definitely something that I am excited that I get to continue at DISL, working with interns and students as part of my job. So by this point, I knew that I wanted to go beyond just working as a biologist or a veterinarian, and that I really wanted to do interdisciplinary collaborative research. So I started applying for dual Ph.D. DVM programs and ended up back in Georgia at the University of Georgia. And there I worked with Dr. Sonia Hernandez for my PhD, and I had opportunities to work in many settings, including the clinic and the field, as well as the lab. And my PhD program is something that I want to highlight a little bit here, the Integrative Conservation, or ICON program, because it's one of only a handful of programs in the country that emphasizes the importance of interdisciplinary collaboration in solving today's socio-ecological challenges. So as a student in the ICON program, you are trained in depth in a home discipline, and then you are trained to get some disciplinary breadth in other disciplines. So my home discipline was the Forestry and Natural Resources School, and that was considered the natural sciences along with ecology. And then I was cross-trained in the social sciences, including anthropology and geography. So my dissertation research had components of both natural and social sciences. And this is something that still heavily informs the way that I approach research questions and ask questions of wildlife health significance from both the wildlife and human standpoint. So as I mentioned, I had both a natural and social science side to my dissertation research, and I focused on the effect of diet on green sea turtle health and nutrition, and the effectiveness of the education programs that are offered at a rehabilitation center, the Georgia Sea Turtle Center. And throughout my doctoral work, I built a large network of collaborations, and I would be remiss if I didn't acknowledge them. So we built collaborations across public, private, and nonprofit institutions that are listed here. And my funding sources are also acknowledged on the right. Green sea turtles are found throughout temperate waters, and they face numerous challenges, including disease, anthropogenic impacts, as well as things like climate change. And for this reason, they are globally listed as endangered by the IUCN. The map you can see below is their distinct population segments as defined by the Endangered Species Act, where they are listed as either threatened or endangered throughout their range. Green sea turtles have a unique feeding ecology in that as pelagic juveniles, they live out in the open ocean, and they are primarily carnivorous. And once they reach a size of about 20 to 25 centimeters, they recruit to coastal waters. They live out the rest of their lives and switch to a primarily herbivorous diet of seagrass and algae. This is important because when animals present to rehabilitation facilities, they are typically sick and debilitated, and places often start out by feeding them an omnivorous diet of a mixture of seafood, including things like herring and squid and shrimp, as well as terrestrial vegetables, such as cucumbers and lettuce and peppers. And importantly, when sea turtles first come in, they seem to really prefer the seafood, which is, I sort of liken it to humans. When we're sick, we don't want to eat healthy things. We want to eat things that are bad for us. But anecdotally, we have noticed that if they're fed seafood for long periods of time, they will eventually become obese and develop problems such as fatty liver disease. So because of that, we do try to switch them over to a vegetable-based diet over time in rehab. But up until this point, up until my dissertation work, all of this has been anecdotal, and no one had really set out to scientifically look at the consequences of feeding green sea turtles a primarily fish-based diet. So I have two studies that I want to share with you from my dissertation work, where we looked at the effect of shifting individuals from that primarily seafood-based diet to a primarily vegetable-based diet. And in the first, we looked at the effect on blood biochemistry and nutritional parameters. And in the second, we looked at effects on the fecal microbiome. I carried this work out in two places. All of the rehabilitation animal samples came from the Georgia Sea Turtle Center, which is a rehab hospital on Jekyll Island. It's just one of the barrier islands off the coast of Georgia. And for the blood parameter study, we had a free-ranging control population that we sampled off the coast of Florida. And as I mentioned, we sampled rehabilitation animals, and we did this at 3 time points: admission, which was within a week of arrival and when they were eating less than 25% vegetables; mid-rehabilitation, when they were eating at least 25% vegetables for at least 2 weeks; and recovery, when they were eating at least 50% vegetables for at least 2 weeks, and they were also considered clinically recovered. And I compared these animals over time, all the clinical parameters over time throughout rehabilitation, using linear mixed effects models. And then for our wild animals, we had 34 of them, presumably eating a diet of seagrass and algae. And we compared these animals to those at admission in rehab and those at recovery in rehab. So for the blood work study, we collected blood from the external jugular vein or the dorsal cervical sinus, and we assessed a wide range of parameters to try and get a comprehensive view of their nutritional status at each time point. And for today, I'm just going to focus on a couple of them that I found particularly interesting. And the charts on the next couple of slides are all going to look similar, so I'll explain the first one in a little more detail. So triglycerides is the first one. That's one of my favorites to look at because we can really relate to it in human medicine. And you can see the chart is set up so that the parameter we're looking at is on the y-axis, and the x-axis has the 3 time points throughout rehab in different shades of blue, and then the wild animals are in green. And you can see the dotted lines, those are identifying the normal reference range identified from the literature. So admission animals, you can see were not significantly different from wild animals in their triglyceride level, but by mid-rehabilitation, after they're eating that highly seafood-based diet, their triglycerides nearly triple, which you can imagine is not great for them. This did decrease by recovery, however, it was still significantly higher, so they were still experiencing residual effects from that high-fat seafood-based diet. Calcium phosphorus levels, they tend to be related reciprocally, so we often look at them as a ratio. And in green turtles, their normal is about 1 to 1. And when calcium drops too low, or calcium or phosphorus becomes too high, and that overall ratio is low, your body will start to pull calcium from the bones to keep circulating levels high. And this ultimately leads to a weakening of the bone integrity and can lead to pathologic fractures. And here you can see that admission animals were significantly lower than wild animals, likely because of that debilitated status when they come in. And their ratio did not increase over time throughout rehabilitation, even though they were actually receiving a calcium supplement during rehab. And we think this is likely because that seafood-based diet is very high in phosphorus. Things like squid and herring have an inverted calcium phosphorus ratio where they're very high in phosphorus. So this is something that is definitely a detrimental aspect of eating a lot of seafood. Vitamin D actually pairs very nicely with calcium, and you can see that throughout time in rehabilitation, it was also significantly lower than in wild animals and pretty much stayed that way, even though they were also supplemented with a vitamin D tablet. And the interesting part about this is that we figured out that they are maintained indoors at the Georgia Sea Turtle Center. And while they have skylights that make it appear like they're getting sunlight, the skylights are actually not UVB penetrating skylights. And your skin, to convert vitamin D, needs UVB rays. So we took an individual turtle who was not in our study and rotated it outside to get natural sunlight exposure. And that animal's vitamin D levels actually tripled in about a month time. So this was something really cool that we were able to share with other rehab facilities, that if possible, you should start rotating your turtles outside and make sure they have access to natural sunlight. And the last one that I want to show you is magnesium. And this is something that came as actually a bit of a surprise to us. Compared to wild animals, admission animals were significantly lower, and this only decreased throughout time in rehabilitation, which is really not great because magnesium is a super important cofactor in a lot of reactions in the body. And it's something that is not included actually on a normal point of care analyzer that veterinarians often use when they look at blood work in critical animals. So this is something that you have to send out for a special test, and that's the only reason we found it, because we were looking at such a comprehensive panel of blood work. So this is something that we were also able to share with other facilities and recommend supplementation. I think humans can really relate to is the microbiome aspect of my project. And we know of the microbiome in humans with their skin microbiome, the gut microbiome, and many other things. And research has shown that our microbiome really plays critical roles in health issues, things from inflammatory disease to obesity, and even things like diabetes. So we know that the diet that we eat influences our gut microbial diversity. So we were really interested in how the green sea turtle gut microbiota is influenced by what they eat. So we collected feces at the same 3 time points that we collected blood from. And I took samples down to the University of Florida and worked with Dr. Tom Waltzik's lab there to extract, amplify, and sequence all the 16S rRNA, or the bacterial genes, from our samples. And the downstream data was analyzed by collaborators at Texas A&M. And here you can see the principal coordinate analysis at the various time points during rehabilitation. So you have red is the admission samples, blue were mid-rehab, and the gold are the recovery samples. And you can see that admission samples were significantly different than mid-rehab and recovery, and mid-rehab and recovery were not significantly different from one another. So this makes sense that admission sample is likely reflective of what they were eating prior to admission, and then the mid-rehab and recovery samples are more reflective of that seafood-based diet offered early in rehabilitation. So the next logical question is, okay, so the compositions are different, but what bacteria are actually driving these differences? And here you can see an overall sort of 10,000-foot view at the phylum level, where admission samples were predominantly made up of Firmicutes, and this gradually changed over time to be replaced by primarily Bacteroidetes. And this also follows our logic in that Firmicutes are better known for their ability to break down plant polysaccharides, while Bacteroidetes are actually better known for their bile tolerance and their ability to break down fat in high protein-based diets. So the importance here is that at recovery, these animals are considered clinically recovered, and yet their gut microbiome is still primarily Bacteroidetes, which means they're primed to be eating seafood and not the seagrass and algae diets that they should be eating. So with this research, we showed that the seafood-based diet does have many impacts, both on the nutritional biochemistry as well as the fecal microbiome. And because of this, we recommend shifting animals to an herbivorous diet as soon as possible after admission. There was always anecdotal evidence that we needed to do this, but now there's some science behind it and that we need to do it as quickly as possible. The final part of my PhD research that I wanted to share is the human dimensions research. And this photo demonstrates right here, demonstrates how the Georgia Sea Turtle Center makes conservation personal by visitors by providing them up-close encounters with sea turtles and giving daily educational programs. So we developed a survey instrument to look at the effectiveness of these programs. And specifically, we looked at the effectiveness of a behind-the-scenes program. So the images here, the left one shows you the gallery at the Sea Turtle Center. And this is a gallery with information exhibits that everyone has access to who comes to the Sea Turtle Center. And in the back, you can see a window, and that's much like the window at the museum that I used to work at, where people can look inside and see what's going on the veterinary side of things. However, there's not a microphone there. On the right-hand side, you can see the behind-the-scenes tour, and this is a special hour-long tour with an educator in small groups that you pay a little extra for, and you are taken to all the areas that you can't see as an exhibit visitor. And you also get to meet the veterinarian and see treatments up close and personal. So I developed and validated a survey to compare these two groups. And in the final analysis, we had 4 constructs, which are listed here. The number of items are the number of questions within each construct. And then the Cronbach alpha, which is a measure of internal consistency and reliability of your questions, is also listed, and a level of about 0.7 is considered good for social science, and you can see that we exceeded that on most of them. So I'm going to focus on the third construct, or the importance of sea turtle rehabilitation. These are the questions for that construct. There were 12 of them, and you can see that they ranged from, on a Likert scale, from 1 to 5, of strongly disagree to strongly agree. And this is a few of those questions called out. So these are the summary results across everyone that I surveyed. So we lumped together here the gallery and behind the scenes. And you can see that over 90% of people either strongly agree or agree. For example, rehabilitating sea turtles is important. That's not really surprising because I'm surveying people who took the time and energy to come to a sea turtle rehabilitation center. So it makes sense that most people there would think that rehabilitating sea turtles is important. And then when you actually collapse that category down and divide the two groups by the gallery and the behind the scenes, you can actually see that the behind the scenes people were overall more likely to agree that rehabilitating sea turtles is important. So this research contributed to our understanding of public attitudes towards sea turtle conservation, and I was able to generate a survey instrument that can actually be pretty easily adapted to other situations. So I envision potentially being able to use a similar instrument here at DISL in many potential capacities. So after completing my PhD, I continued to build on my wildlife health research experiences, and I worked as a research technician with the Southeastern Cooperative Wildlife Disease Study under Dr. Danny Mead for about 3 years during vet school. And for anyone who's not familiar with SQUIDIS, as they call themselves. It's a diagnostic lab based at the University of Georgia. And they're called a cooperative because it's made up of about 20 member states that actually pay to be members. And as members, they get free diagnostics on all wildlife that are submitted from their state. So in the lab that I worked in, we worked primarily on active and passive surveillance for West Nile virus in both mosquitoes and birds, primarily using molecular tools like virus isolation and qPCR and conventional PCR. And we also worked on emerging fungal pathogens like snake fungal disease and white nose syndrome and chytrid. And I developed and maintained several qPCRs for those. And I also had the opportunity to work on necropsies of a lot of various interesting wildlife because they came from all over the US. So that was something really exciting. I also completed several rotations during vet school, and one of them was with the California Department of Fish and Wildlife. And I'll see if this video wants to play. It might be a little slow because our internet here isn't great, but hopefully you can see that there's a helicopter, and suspended from that are 4 mule deer in California. And the helicopter crew would go out and net the mule deer. They are hobbled and blindfolded for their ride below the helicopter and brought back to a base camp where we would weigh them, which you can see in the left picture here, and then take various samples such as blood as well as hair and feces. And we would also use ultrasound to check them for pregnancy. I also had the opportunity to work with the Marine Wildlife Veterinary Care and Research Center, as well as the Lindsay Wildlife Museum. So this was a really great rotation that I did actually fairly recently. It was in 2020. I was also fortunate during vet school to receive a Boehringer Ingelheim Summer Scholar Award and travel to the Netherlands to work with Helena Verhey on an infectious bronchitis virus project. So infectious bronchitis virus is a highly contagious respiratory disease in poultry, and it causes significant economic concerns because the illness can lead to decreased egg production and quality. And the Netherlands, much like the US, eats a lot of chickens. So it was an important project. And we were looking at the differences in replication and pathogenicity of the virus in a virulent genotype of the virus compared to a live attenuated vaccine. And we used PCR as well as immunohistochemistry, which you can see in the image here. This is actually I believe it's the cloaca, and the blue cells are normal, and the red cells are areas where the IHC or immunohistochemistry is actually highlighting the viral antigen. So this is a case that was positive for infectious bronchitis virus. And we found in this study that the attenuated phenotype does show slower dissemination and lower replication throughout the body, which is not surprising, but it needed to be proved. So now that it has been published, which is awesome. And then the final thing that I want to share with you from my veterinary time is an externship that I completed with the Michigan Wildlife Disease Lab. And I actually was able to conduct research with them in collaboration with Michigan State University on chronic wasting disease. So chronic wasting disease, for anyone who's not familiar, it's not present in Alabama, so it might be new to you. It's a transmissible spongiform encephalopathy, much like bovine spongiform encephalopathy or mad cow disease. And it is caused by a prion, which is an infectious protein. It affects a wide range of cervid species, and it's a progressive disease that results in clinical signs that you can see here. And it's actually, unfortunately, it's 100% fatal once these clinical signs develop. The diagnosis for chronic wasting disease is via the retropharyngeal lymph nodes. So the best diagnostic tool that we have is postmortem, unfortunately. And typically, because of cost and ease, there's a screening test that's done with an ELISA, and then a confirmatory test, which is done with immunohistochemistry, much similar to what I was sharing with infectious bronchitis virus. So for this project, I looked at white-tailed deer that were positive via ELISA, so that initial step, and then I compared the immunohistochemistry slides from the same animal. And I was interested in how often the IHC, or the confirmatory test, differs from that of the ELISA. And in addition, many agencies on the ground will only collect one of the two available lymph nodes for analysis. So I was interested in how often the two lymph nodes from the same animal differ. And here you can see that in 87% of cases, both lymph nodes were positive via IHC. So in 87% of the time, they agreed. And then in almost 7% of the time, the lymph nodes were actually negative by IHC. And also, very importantly, in almost 6% of time, one lymph node was positive and one lymph node was negative by IHC. And there's many reasons that IHC might be different, the results, than ELISA, but it's something that's very important to point out to agency personnel, especially that fact that you should probably collect both lymph nodes if you want to increase your sensitivity of finding CWD. So in the last couple minutes, I just want to take a minute to talk about my position here at DISL and what I've been doing since July. So here you can see that one major part of my job is helping with necropsies and live animal stranding response. And so far this year, as of yesterday, we have 58 bottlenose or suspect bottlenose dolphin strandings. And hopefully this video plays for you. I'm sorry if it's slow again, but you can see that a necropsy is a big event. Lots of people are there to aid. And because of coronavirus, though, we've actually been doing about 10 necropsies since I started, all with limited personnel, about 4 to 5 people. So they do take quite a bit of time. That was a time-lapse video. They're not normally that fast. So part of my job is the necropsy. And then the other part of my job is research. So I wanted to share one short thing about what we're working on with some stranding demographics. So the who, what, when, where, and why of dolphin strandings in Alabama. And here is a chart showing the number of strandings per year on the y-axis, and then it's broken down by size class on the x-axis. And the triangles in the image are highlighting the 2011 unusual mortality event, or UME. And these triangles, you can see, they really emphasize the disproportionate number of fetuses that stranded during that year, which was actually ultimately linked to late-term pregnancy failure in the dams due to poor health following the Deepwater Horizon oil spill, as well as potential brucellosis infection. And while there were no significant differences in the number of strandings by size class, we do see a trend here in that the larger size classes are stranding, or at least being found, more often. So whether they truly die more often due to old age or disease, or maybe being more adventurous and likely to run into trouble, as the cases of subadults, or it could be that we find these larger size classes more often because they persist longer in the environment or are more easily found by the public. So along those lines, we are also looking into cause of death in these individuals, and as well as infectious disease prevalence like brucellosis and morbillivirus. I am also particularly interested in continuing working with the microbiome, and there is work out there on the dolphin skin microbiome, but we're looking into some novel sampling techniques for being able to look at the dolphin skin microbiome, and as well as no one has yet looked at how their skin microbiome changes with exposure to fresh water, which is a huge problem in the Gulf of Mexico here, or how that microbiome changes with decomposition. So what we can infer from a swab of the skin based on how long the animal has been dead. I'm also very interested in the pathogenesis of the freshwater skin lesions, which you can see in this dolphin here. These lesions are a result of that animal spending time in water of low salinity. And also in necropsy, we take tons and tons of subsamples of every organ you can imagine. And we work with a lot of different labs, both externally and internally. And one of the collaborations that I want to highlight here is with Dr. Allison Robertson's lab. And that's an internal collaboration here at DISL. And it's something that we are really interested in expanding if anyone has ideas that they would like to work with us on. So to wrap up here, my research vision really ties back into my perspectives on wildlife health research, and I hope to be able to share and integrate my veterinary and wildlife biology skills. In addition, I truly believe that humans play a critical role in animal health, and that's not limited to dolphins, but definitely with dolphins and marine mammals in general, and also with stranding response. So I'm interested in potentially starting some human dimensions research. And finally, I've mentioned many times that I'm a strong believer in collaboration. So if anyone was intrigued by some of the things that we're doing or could do, please feel free to contact me. And my email is included here. And this is my husband, Matt, whose name has taken over my Zoom account, and our dogs, Blue and Zoe. And I'll take any questions.
With the change of the gut biome in sea turtles and correlating it a little bit with data showing that in dogs and cats with antibiotic use that it can take months for the gut biome to increase, is other than increasing the or starting the vegetables earlier, would there be any indications for extending that recovery period to allow that gut biome to maybe get closer to normal?
Absolutely, that's a great question and a really important thing to point out. And a lot of our individual turtles actually did receive antibiotics, so that's not something that I shared, but it's something that we looked at. We did not see a significant change in their bacterial diversity due to antibiotic use, but it's something that's absolutely definitely been documented because for anyone who's not familiar, the antibiotic will basically wipe out your gut flora, and you're starting sort of from a baseline and rebuilding. And a lot of your gut microbiome, especially with oral antibiotics, will take a long time to rebound. And so it's something that we definitely looked at, and it's a really good thing to consider. And I think that could have played a role in our study, even though we didn't find a significant decrease in the diversity or composition of the bacteria. And I agree, yes, that extending sort of the time that they're in rehab, not only because of the antibiotic, but also because of the obvious shift due to diet, would be a really important thing. I think that a healthy gut microbiome is one that is really diverse in the number and composition of the bacteria that they have, and that would theoretically only improve with time.
Thank you.
Any other questions? Well, thank you everyone so much for coming, and feel free if you see me in the hallways to introduce yourself. I'd love to meet some new people.
Hi, Jenny. There are 2 questions in the chat window.
Oh, okay. I can't see that for some reason. Let's see. Is it possible for you to read them? I'm really sorry, I can't.
Okay. So there's one audience from Discovery Hall. They are asking, are you planning to do anything with the social science research here at DISL?
That's a great question. I would really love to. And if that's something that I haven't looked into how much you guys probably already do with your programs there, but if you're interested in collaborating on something, I would love to do that. It's something that I'm very interested in. I think that education programs play a critical role in people's attitudes and perceptions about conservation. So it's something that I am pretty passionate about, and I would love to collaborate on anything if you guys want.
All right. I think that's it. If later audience came and missed some content, this video will be available on the YouTube channel at the DISL. So feel free to pull them out. And if you have any questions, you're welcome to leave any comments under the video as well. So with that, if there are no any more questions, let's call it a day today. Thanks, everybody. Thanks, Dr. Bloodgood.
Thank you, Sean. Thanks, everybody.